Hello, everyone. My name is Shalyn McCoy, and on behalf of the Atlantic Salmon Conservation Foundation, I would like to welcome you to today's webinar. The series is made possible in part thanks to the contribution of the Government of Canada. For today's webinar, we are pleased to be hosting Dr. Xavier Bordeleau. Dr. Bordeleau has completed his PhD at Dalhousie University, Nova Scotia in 2019, working on the post-spawning ecology of Atlantic salmon in eastern Canada and anadromous brown trout in Norway and sub-Antarctic Kerguelen archipelago, and will be presenting on this work today. He is now a Pinnip research scientist at DFO's Maurice Lamontagne Institute, working on the spatial and trophic ecology of seals as well as predator-prey relationships. Today, he'll be speaking on the post-spawning ecology of iteroparous salmonids, basis of variability in migratory behavior and survival, ecological importance and conservation implications. So after this presentation, we will give an opportunity for folks to do questions and answers, and I'll explain at the end of the presentation how we're going to go about to do that. Now it's time to turn the webinar over to Dr. Xavier Bordeleau for his presentation. So Xavier, it's yours.
Okay, thank you very much. Hi everyone, I'm Xavier Bordeleau. I'm a research scientist at the Maurice Lamontagne Institute, so at DFO in Quebec. And as Shalyn mentioned, I've transited research mandate about 3 years ago, and now focusing on pinniped ecology and predator-prey relationships. But of course, salmonids still holds a very special place in my heart, and so that's why I was glad to be invited to this webinar series, and mostly present on my PhD research at Dalhousie University and following up work. And so I'll be talking about the post-spawning ecology of iteroparous in relation to their migratory behavior, survival, the ecological importance of repeat spawners, as well as associated conservation implications. And so my PhD journey first started with work on endangered Atlantic salmon populations in eastern Cape Breton, eastern Canada, to document some of the causes and consequences of individual variability in behavior and survival of kelts. And then this expanded to addressing similar research questions, but in another related species, but across different population contexts. So working on native anadromous brown trout population in Norway, which eventually led to following up work outside the aims of my PhD, still in Norway, but also working on introduced brown trout population in sub-Antarctic waters to study the marine habitat use and feeding ecology of introduced brown trouts in these waters. But for the purpose of today's talk, I'll mostly focus on 2 of my PhD chapters. So the first one, basically looking at the importance of iteroparity across Atlantic salmon population of the Northwest Atlantic, and the second one looking at some of the consequences of current hatchery practices for wild broodstock. And then, as I quickly mentioned, interesting opportunities came up to work on marine mammals at DFO. And while fishes have been my first love in research, seal eat fish and get eaten by fish. So in addition to be quite fascinating creatures themselves, so I'll be talking or discussing more briefly my current research program, and also mention some collaborative ideas linking back to salmonids, which include the current project in Denmark, looking at harbor seal predation on anadromous brown trouts in a fjord system over there. But before doing so, I'd like to acknowledge the important contributions of all my colleagues who all very significantly contributed to my current expertise and background, and especially to my supervisor, Glenn Crossin, who I've had the immense pleasure of working with for 5 years in Halifax. And also a special mention to Jeff Hutchings, who very sadly passed away last year. And Jeff was such a great inspiration to myself and many others for his devotion to science and science rigor. So I suspect this audience is very familiar with this, but so that everyone are on the same page, here's a general life history scheme of salmonids who first hatched and developed in freshwater before going through the process of smoltification, consisting in many physiological adaptation to be able to tolerate salt water. At which point they can migrate to sea, reach productive feeding grounds, grow before returning to their natal river to spawn a single time before dying. So growing up, at least, that was the salmonid life history scheme I learned about. Through a nature documentary that were primarily or almost entirely focusing at the time on ecological Pacific salmon species. However, Atlantic salmon, brown trout, and in fact many other salmonid species are iteroparous and can reproduce multiple times in their lifespan, which means that there's also a whole other life history stage to learn about and consider. So while they have the capacity to survive the first reproduction, the energy investment is such that after spawning in late fall, kelts are found in a depleted nutritional state at a particularly difficult time of the year. And so as such, the post-spawning mortality can be quite substantial. And for that reason, the contribution of repeat spawners has mostly been overlooked. And they're not always accounted for in current management practices. A basic example of that would be in the design of downstream dam passages that are constructed and designed for small juvenile fishes like the smolts, but not necessarily for large-bodied post-spawn adults or kelts that would also be migrating downriver at the same time. And so despite their relatively low representation in annual return, at least for Atlantic salmon, repeat spawners are experienced spawners of large body size, which also translates to high fecundity in females. However, despite their potential importance for population viability, still relatively little is known about the kelt life stage. And this is still the case because that citation is a bit dated. But this is still the case and especially true as it relates to the factors influencing the movement ecology and survival of post-spawners in freshwater, estuaries, and at sea. So to address these knowledge gap and better understand the potential causes of differential migratory decision and success, I combine acoustic telemetry and physiological sampling to look at how endogenous factors in different species, in different ecological contexts, or other anthropogenic activities are then influencing the migratory behavior of animals and their survival. So here, I think people are mostly familiar with some of these methods, but I wanted to highlight what we've done. And so fish were captured in the rivers at the tagging location. In Kerguelen, it was the estuary, also in Norway. The main idea is that we use angling as the primary capture method to individually deal with each fish. And that was very important when we get at baseline stress parameters so that every fish could be sampled within a maximum of 3 to 5 minutes after the initial stressor was perceived. And so this is what it's showing in the Kerguelen Islands. A similar methodology again in Cape Breton in Norway. And so once blood samples were collected, fish were anesthetized, and then in Cape Breton and Norway, we then proceeded to surgically implanting acoustic tags in the body cavity, and then the wound would be sutured up while the gills were being continuously irrigated to make sure the fish was breathing and getting the oxygen it needed. The difference in Kerguelen is that we wanted to use tags that would have salinity and temperature sensors to inform us about the environmental conditions that these fish encountered. And so the acoustic tags were mounted externally, as we can see there. And then scales were collected for aging and spawning history. Fish were weighted and measured before really being released back in their environment. And so acoustically tagged fish are then back in their environment, and then we would deploy acoustic receivers to form arrays. Often these can be deployed in series of gates to cross off narrower passages in some of the systems. So we can see the array map of the Norway systems I worked in there. So while I had just started off on this tagging and physiology work, Jeff Hutchings, who was an external examiner in my proposal defense, had a simple but still extremely relevant question that I thought needed to be addressed first, which was basically around why should we care about the movement ecology of kelts if most of them died anyway? Or the way we then decided to look at it, what is the role of iteroparity or repeat spawners for population viability? And so specifically, we wanted to look at certain aspects, such as what proportion of annual returns are represented by repeat spawners, and how did that change through time and space? Also, how much do repeat spawners contribute to in terms of total annual egg deposition? And can they represent a safeguard against low recruitment periods? And so I began to look for answers about the ecological role of iteroparity, but the reality was that there wasn't much out there in the available literature. So I was quite perplexed by this, and this led me, in fact, to a multi-year quest for data in setting up the basis that would make this study possible. And so from the start, I want to acknowledge the vital contribution of colleagues who kindly accepted to share their data and allow me and us to address these important questions. So Gerald Chaput and Ross Jones, Brian Dempson, Martha Robertson, Alex Levy at DFO, as well as Julien April in MFFP Quebec, in addition to published data. And so thanks to everyone's contribution, I was able to assemble 11 population time series of annual return, which are highlighted in the map below, which span from 21 to 47 years. And the time series consisted in information on the spawning history composition of annual returns. And we decided for the purpose of this study to summarize this in 3 groups. So the first 2 groups are maiden or first-time spawners, so either 1C winterfish that have a more local migratory strategy, or 2C winterfish and 3C winterfish that would be migrating off the coast of West Greenland. And then for repeat spawners, we could not distinguish consecutive and alternate repeat spawners, so they were summarized as one category. So despite the high variability in the representation of repeat spawner among population and years, we did identify a spatial coherence in the temporal trends of iteroparity across population of Atlantic salmon in the Northwest Atlantic. So this was done using a dynamic factor analysis, in which we considered various configuration and various number of common trend shared amongst population. Interestingly, the most parsimonious model when considering all population together identify a single common trend shared amongst time series, but the loadings on that trend seem to be correlated spatially. And in fact, that was showing that mid-latitudinal and northernmost population exhibited an increase in iteroparity, so with positive loadings on that trend, and the specific trend generated for these population alone as being displayed. But basically, iteroparity more than doubled during that period of time in many of the populations. While we were seeing negative loadings on that trend in southernmost population, showing a decline in iteroparity over the past few decades. And so following that, for 4 populations for which we had data on sex and size, we were then able to calculate the proportion of eggs contributed to by repeat spawners. So first of all, looking at the spawning composition for female alone, and then based on fecundity length relationships for specific to this population, we're able to extrapolate the egg deposition, so the reproductive contributions of these different groups of fish. And because repeat spawners are mostly constituted by females of larger size, an overall 9.5% of repeat spawners in the annual return of sex combined contributed to about 1/5 or 19% of all eggs being deposited. And finally, we wanted to look at the idea that repeat spawners might be acting as a safeguard against years of low maiden returns. And to test that hypothesis, we were not interested in the overall trends, which we just discussed, but more so in the shorter term fluctuations and the relationship between these. And so we isolated the high frequency variability component by detrending these time series to look at the relationship between the counts of maiden spawners and the proportion represented by repeat spawners for these years. And this indeed revealed that there was a generally higher representation of repeat spawners in bad years, or the years where the counts of maiden spawners or first-time spawners were at the lowest. And so not only that repeat spawners can contribute to a significant proportion of all eggs being deposited in a year, but that they do so when a population needs it the most. So these are just a few examples of the really the poorer or lower return years in the Trinity and Miramichi River, and how much repeat spawners contributed in terms of total egg deposition. So we could see that this spanned from about 27% to up to 42% of all eggs being deposited in these years of what could be interpreted as low recruitment. And so despite high variability in the representation of repeat spawners, we did identify broad-scale temporal shifts in iteroparity with increases in mid-latitudinal and northern population, in part associated with the timing of fisheries closure for Atlantic salmon, commercial fishery closures. This highlighted that there is a potential for iteroparity to increase when some anthropogenic threats are mitigated, with quantified benefits to population resilience, notably their important reproductive contribution and safeguard against low recruitment period. And while iteroparity is an important bed hedging strategy, allowing adults to spread the risk of reproductive failure over time, the steep decline in juvenile survival observed in southern population, along with reduced post-spawning survival due to anthropogenic threats in freshwater, is of particular concern for population resilience. And so these findings were recently published in ICES Journal of Marine Sciences. So considering the importance of iteroparity for Atlantic population, I would then argue that it is in fact important to study the factors that are influencing the migratory behavior and success of kelts, which then bring me to a brief description of my second thesis chapter, looking at the nutritional correlates of overwintering and seawater migratory decision and longer-term survival of Atlantic salmon kelts. And so to tackle this question, we then evaluated the influence of nutritional state on the post-spawning migratory decision and success. So we looked at the downstream river migration timing, the overwinter mortality, the estuarine residency of fish, as well as their marine progression and longer-term survival to come back to spawn. So to do so, the study was conducted in the Bordeleau Lakes in the Middle River, where we tagged 25 kelts in fall 2015, which we tracked for more than 650 kilometers. And as they're making their way from the Middle River to the Bordeleau Lakes, as we can see on the map, they would cross a series of gates to reach the Atlantic Ocean, either to the Great Bordeleau Channel or Little Bordeleau Channel. And then once they're out in the Atlantic Ocean, they would cross the Cabot Strait to enter the Gulf of St. Lawrence, and eventually the Strait of Belle Isle for those that are heading towards the Labrador Sea. And I also conducted lab assays for plasma triglyceride levels, which is the main form of stored energy in animals. And so overall, kelts in lower nutritional condition, so lower plasma triglyceride level and lower body condition index, showed earlier downriver movement. So using the tidal part of the river system more extensively during winter, which might yield more feeding opportunities with the presence of smelts, for example, for those animals that cannot afford to starve an extra few months higher up in the river. But that came to a greater risk of overwinter mortality, presumably due to a higher predation risk in that area, but also the fact that they were more nutritionally depleted. Once they moved out of that large estuarine habitat formed by the Bordeleau Lakes, these lower condition animals spent more time reconditioning in the estuary, presumably due to higher nutritional needs prior to their oceanic migration. And ultimately, these animals exhibited lower repeat spawning, survival to repeat spawning. So overall, these findings indicated that differences in nutritional state underlie spatiotemporal variation in the habitat use of these fish, and that repeat spawning potential was at least to some extent limited by nutritional constraints. So then we had the opportunity to work with the Marguerite Provincial Hatchery to look at how hatchery spawned kelts compared to their naturally spawned counterparts when they were released back to their natal rivers. And so broodstock collection and enhancement programs are widely used conservation practice throughout the species global range. And often these programs consist in the capture of wild adult Atlantic salmon that are coming back up the river to spawn, at which point they're intercepted by the use of seam nets or counting fences. These fish are then transported back to the hatchery, retained in captivity until sexual maturity. So that can spend from 1 to 6 months, depending on programs. At that point, they're stripped from eggs and sperm and will later be released back to the river of origin. And so, as we can imagine, such practices present many stressors for these fish. So from the initial capture event to transportation, frequent handling, air exposure, as well as the effective captivity itself. And while significant effort has been directed at improving rearing condition for juveniles salmonids, the impact of captive spawning on the future reproduction of iteroparous broodstock had received minimal attention. And that's especially relevant considering that they can make important reproductive contributions and with the patterns that I discussed in some of these areas. And so one important concept to this research is the stress response. So I'll briefly introduce this. Once a stressor is perceived, such as captivity or handling, the primary stress response involves the activation of the HPA axis, which leads to the secretion of cortisol, the primary stress hormone. And following cortisol secretion, the secondary stress response then involves some metabolic changes. And one major effect is an increase in circulating glucose level that is basically used to power behavioral responses or organismal level changes that are necessary to cope with the stress. And so this adaptive stress response, which causes the temporary suppression of other physiological mechanism, can also become maladaptive if a stressor is sustained over a long period of time, and so moving to chronic stress, eventually affecting individual fitness. So to evaluate the effect of hatchery breeding practices on the repeat spawning potential of wild broodstock, we assessed differences between hatchery and wild spawn in relation to the following aspects of their stress level, but also their immune state, their migratory behavior, and their longer-term survival. So by combining acoustic telemetry and physiological sampling, we aim to link the different levels of impact that hatchery might have on the future reproduction of adult salmon. Sorry, I'm just lots of speaking. So first, looking at the pre-migratory physiological state, we could see that both stress indicators were by far elevated in hatchery spawn kelts, either through plasma cortisol or plasma glucose. And we've also quantified the circulating concentration of a compound, prostaglandin E2, which is involved in the inflammatory response, which was previously associated with the inhibition of several immune-related genes. I also want to point out that there were no differences in morphology, length, weight, and body condition of these two groups of fish. So now looking at their migratory behavior from the initial group of kelp that were either tagged at the hatchery before being released or tagged in their natal river in the same period, a similar proportion of each group survived through the freshwater stage to make it to the river mouth and enter the estuary. And while there were no differences in overall freshwater survival, the timing at which fish left the river to enter the estuary differed markedly between these two groups. So in fact, hatchery spawn kelts exited the rivers prematurely in many cases, and in an average 66 days earlier. And so while all wild spawn kelts migrated through the estuary in the spring, about half of the hatchery spawn kelts did so in the wintertime. So from these similar proportion of fish that made it through the estuary, then all the wild spawn kelts survived to the Atlantic Ocean, and estuarine survival was lower for hatchery spawn kelts. So this is what we can see here. Evaluating the different factors that might be contributing to the different survival patterns between these group of fish. The most influential single predictor of estuarine survival for hatchery fish was in fact the timing at which they left the river. And so from what we can see, it's really those early hatchery migrants that had very poor survival. So only 33% estuarine survival for these animals. And so, as I mentioned, we were able to track fish through their entire oceanic migration. And so none of the hatchery fish made it back to the Nato River, while 6.5% of well-spawned kelps, which is 9.1% just looking at well-spawned females, which is consistent with scale reading estimate for that population, in which iteroparity tends to be low at around 5%. And so collectively, hatchery spawned kelps differed to well-spawned kelps counterpart on many different levels. Physiologically, they showed high stress levels and potentially alternate immunity. And behaviorally, these animals left the river prematurely, for which these early hatchery migrants then to a greater extent once out in the estuaries. And while these are certainly low sample sizes, ultimately none of the hatchery spawned kelps contributed to future generations. So overall, these findings indicated that hatchery practice can affect individual fitness with potential level consequences, potential population level consequences, depending on the repeat spawning rate in population, but also the size of hatchery programs. And what I mean by size is the proportion of spawners that are spawned in captivity and the proportion that is allowed to spawn naturally. In case of the Margaree River, it is only a minor fraction of fish that are collected, but in other broodstock collection program, like some population in the US, like the Penobscot River is 95% of the spawning run that is brought back to the hatchery, if I'm not mistaken. And so this emphasized that repeat spawning potential should be considered in hatchery programs, and that actions to mitigate the many stressors that they face should be envisioned. And that would be beneficial for kelps, of course, but also for eggs and juveniles due to maternal effects. So the effects, the stress of the parents, the female that is passed on to their eggs, affecting survival of the offspring. So these are also published in Biological Conservation. So then moving to my last PhD chapter, I then applied a similar approach to that of chapter 2, but in a different but related species, the brown trout, in a different ecological context, documenting the nutritional correlates of spatiotemporal variation in the habitat use of brown trout kelps in Norway. And so the brown trout quickly is quite a fascinating species, because behind the dichotomy of anadromous and freshwater residency, it's also documented that brown trouts are expressing a continuum of different migratory strategies, some remaining only in the estuarine habitat, other doing more shorter marine migrations, and other really long oceanic migrations. And of course, that influence their threat, their level of exposition, to different threats in the coastal environment. But the determinant of this high individual variability are not understood. So for that, I joined forces with Jan Davidsen and Sindre Eldoy to incorporate physiological sampling in the research program and see what more we can learn. So this is the Tolsen Fjord and the array of receivers that was deployed over there. So the objective was to evaluate the influence of nutritional state on the extent of the marine habitat use of brown trout kelps. And so we looked at their migratory tactics, distance and area use, their marine residency period, as well as their marine survival. And so for that, we tagged and biosample 32 kelts in spring 2015, which we then tracked through their summer feeding migration in the fjord system, and conducted a laboratory assay for plasma triglyceride levels. And so here on the right panel is an example of the different marine migratory tactics that are exhibited by these fish. And so we could see some short marine migration, some that we characterize as inner fjord migrants, and other as out of fjord migrants, which are the animals that ventured to the outermost, most pelagic area of the fjord system. So spatially, kelts in lower nutritional condition, either through low plasma triglyceride and low body condition index, adopted further, more pelagic migratory tactic. And temporally, within each migratory groups, depleted individuals spent more time at sea. And these animals in poorer condition also exhibited lower overall marine survival. And so while the sample size are certainly limiting, these findings indicated some nutritional factors underlying spatiotemporal variation in the marine habitat use of brown trout, with depleted individuals generally opting for riskier migratory tactics. Following the observation, then larger, more depleted females were migrating further out and spending more time. This then brings some very intriguing questions for these to be due to potentially due to higher reproductive investment in these animals and a greater reconditioning need. And so the findings were published in CGFAS. This then led to follow up PhD work from my friend and colleague, Sindre Eldoy, who then tested these questions, but over 280 brown trouts that were tagged across 7 different populations in Norway, confirming, amongst many other aspects that he also tackled, but that animals in poor nutritional conditions remain at sea longer, and migrated further offshore compared to fish in better condition. We also found that migratory tactics were highly repeatable in consecutive feeding seasons for those animals that the tag survived long enough to get consecutive behaviors, which really opens up the door for so many different questions about longer term determinants of migratory decisions and tactics, and also their adaptability to changing environmental conditions. And so collectively, this work highlighted that the postponing migratory decision and success of kelts were partly influenced by their physiological state, with nutritionally depleted and stressed animals generally opting for riskier migratory decisions. This suggested that postponing survival is, at least partly, limited by nutritional constraints, but that there's potential for iteroparity to increase when some of these anthropogenic threats are mitigated. With quantified benefits to population resilience, acting as important and making important reproductive contribution, and acting as a safeguard against periods of low recruitment. And so in this context, the potential disappearance of iteroparity is a severe concern for population resilience in some of these areas. And so just to say a few words, because I've been transiting research areas. And so since then, the body of literature continued to grow on KALS. So these are just a few examples of recent paper that I enjoyed reading. Whilst some knowledge gap remains, this growing body of literature allowed colleagues and I from Canada, Norway, and Denmark to write a review of the current state of knowledge on iteroparity in anadromous salmonids, which is currently in revision. And the idea was also to highlight pressing areas of research on the topic. And so I wanted to conclude this seminar with a few words on my current research mandate on pinniped ecology and predator-prey relationships, to highlight some potential collaborative ideas to look at salmonid predation in Atlantic Canada. And so the idea, the general objective of my program is to better understand the role of pinnipeds in their ecosystem and their impact on fisheries. And I want to acknowledge three key people in this effort. The technician working with me, Samuel Mongrain, as well as my predecessor and recently retired Mike Hamill, and Fanny Pelletier, who's a key academic collaborator. So of course, the pinniped research programs involve research on the spatial and behavioral ecology, mainly of gray seals and harbor seals in the St. Lawrence, in the Gulf of St. Lawrence and the St. Lawrence estuary, with many different graduate students who are tackling certain aspects of this. Pierre-Olivier Cusson, who's working on assessing changes in gray seal habitat use of the Gulf, with a series of satellite deployments dating back to 1993. And Sandrine Beaumont-Courteau, who's looking at how dive profiles and dive behavior change over these past 3 decades. Angela Berge is looking at the environmental effect on harbor seals, pup growth, and bird timing in the St. Lawrence estuary. And what I want to get at is that another component of the research program then involve work on diet and trophic ecology. Ali Rani and Théo Garnier, who are 2 students looking at these aspects. And so Theo is contrasting a traditional, so hard parts analysis and still in the seal digestive tracts, and also combining stable isotopes and fatty acids, but recently DNA metabarcoding to develop a more comprehensive understanding of pinniped's diet. I will mention a few words. So of course, there's some potential to look at salmonid predation, although the sampling would need to be biased towards some areas that would be of interest to salmonid researchers. I'll mention a few words about my current collaborative project on spatial temporal interactions and predation of pinnipeds on salmonids in Denmark, and some data coming from a program which aims at identifying a white-shark critical habitat, but also the biological association with gray seals in the Gulf of St. Lawrence. So I'll be very quick because I don't have much time left. So this was evidence of a dinner bell effect. So basically, due to the fact that pinnipeds and many marine mammals can hear at 69 kilohertz, and whether or not this would cause biased estimates of mortality of fish tagged at 69 kilohertz. And so I got in contact with Martin Lee Christensen and Jonas Thielman in Denmark, and so who were both already working on sea trouts and harbor seals in that system. And I thought that would be the perfect opportunity to join forces and tackle these questions. And so we had the aims of comparing survival rates between brown trout tagged at 69 kilohertz, which would be audible to marine mammals and pinnipeds, and 180 kilohertz tags. And also looking at quantifying the spatiotemporal overlaps and interaction between predator and prey. And so tagging is now done. These are the impressive sample sizes. So tagging was done over 2 years, and we are now receivers were actually recovered very recently, and we'll dig into these analysis. But I thought that not necessarily replicating it, but doing something similar in some systems in Atlantic Canada would be of great interest to study relationship between pinnipeds and Atlantic salmon. And I must say that for this effort, all the acoustic array was doubled. So receivers registering 69 kilohertz tags and other at 180 kilohertz. So it was quite a monumental investment, which I'm sure will yield very exciting results. And my very last slide was to say that while the project on white sharks aims at documenting the critical habitat of white sharks in Atlantic Canada, including the water of the Gulf, a secondary aim is also to look at how white shark presence might affect gray seal behavior and look at spatiotemporal overlaps and association between the species. And so for that, we deployed VMTs, which are mobile transceivers. And so we deployed in collaboration with all these great people, deployed some in Kushibuguak, so on the south coast, the east coast of New Brunswick, last summer. And so what we can see with the pink dots are acoustic detections. And so knowing that these are notably close to the Miramichi River, other areas of interest, maybe I was just wanting to pass on the words that over the next few years will generate, of course, white shark detections, but also these gray seals would also detect potential prey, which might be of interest to some folks around the Maritimes. And so this is it. I'd be happy to take any question. And sorry for the struggle. My throat, I had a bit of a hard time speaking like that for an hour nonstop, but I'd be glad to take on some questions. Thanks.
Yeah, you did a great job, Xavier. Thanks for your presentation and for all your work and being able to share the results from that research. So to open it up to questions and answers, there's 2 ways to go about it. You can always type in your question and I can read it out loud for Xavier. Or if you're comfortable and you just want to ask your question by yourself, you can raise your hand and I'll unmute you and you'll be able to ask your question. I see that we already have one with a hand raised. Now let's see if I can unmute. Okay, let's see here.
Can you hear me?
Oh, hello? Perfect. Yes, hello.
Yes.
I was struggling with the unmute. Go ahead.
No problem. Jim Din, I'm from St. John's, Newfoundland, the MHA for St. John's Centre and sort of the, I guess, the fisheries critic as well, in one of my many roles. And I apologize, I didn't get to see all of this because I was called away to respond to some announcements by our government here on a few things. So I had 2 questions. Is it possible to get this presentation as well? And I might have some further questions at the end, but at a later time. But one that I it's certainly here for the last one when you're talking about pinniped seals. I'm just curious, I guess any preliminary research, and I say this from this point of view, it's been a source of discussion in the House of Assembly here in Newfoundland in the terms of the need for a seal cull, because they are basically wiping out, or they're impacting the cod stocks. And I would say that we're getting that from when it comes to probably salmon as well. Now, understand me, I have no issue with a seal hunt or anything along those lines, but I'd rather have it on science as opposed to, you know, like anecdotal observation is important. But I'm just curious as to what you found. My comment in the debate was that a large part of, like, when it came to cod stocks, is that we probably created the situation our own selves through overfishing, and that seals were around when I guess the first Europeans came here as well, and fish stocks were in healthy form. So my question is this: in that context, what have you any hypothesis or expectations or preliminary results with regards to the impact of seals, whether they're harp seals or the I think the gray seals, are on our fish stocks in relation to that, and how they're impacting recovery, growth, or whatever?
Yeah. Well, this is the million-dollar question, isn't it? But also not necessarily specific to salmonids, but yeah, there is a large amount of effort right now, and we're trying to build on it. But to address the many different components that are needed in terms of the data to address these aspects. So one of which is the diet information, which has its own limitation, of course. The other one is the scaling up factors. So right now we're working on even the abundance estimate is based on the distribution of birds, for example, for gray seals. And so we have an ongoing project to try to describe the presence of gray seals across different fisheries management units within the different seasons. And so then with that information, we'd be able to potentially extrapolate some of the diet work for consumption estimates on the different stocks. So I do not have anything preliminary to share, mostly because we're starting a new series of analysis with recent diet data and all these factors of abundance and foraging effort that need to be taken into account. Also due to the fact that my mentor and predecessor, Mike Hamill, has also been working on this issue for a number of decades. But in terms of the new information, we have many projects, as I presented, to try to tackle these questions. In terms of the salmonids, this is a bit more tricky because just randomly sampling, for example, the gray seal population in Central Gulf, like the Magdalene Islands, like we don't find salmonids because their density is such that other preys are far more important for gray seals than salmonids. And so that's why I think a lot could be learned. You just need a few odds or specialized animals to really have an impact on some, presumably some spawning run, for example, or seaward migrating fish. And so I think to answer these specific salmonid question, sampling for seal diet information would really have to be biased towards some areas of interest for the salmonids population. So this is why I was kind of reaching out to people who have interest in these questions, because yeah, this could be interesting to look at.
Okay, thank you. So, and I'm going to finish with this. I saw earlier in the presentation the use, I think, of as a DNA or a genetic. So I'm assuming, if I remember correctly from a presentation months ago or a year ago, is that you can pretty well, if I understand DNA sampling, you can probably figure like what the diet primarily has been of a seal without, let's say, examining the contents of its stomach. That's the first question. And secondly, in relation to the predation of seals and on salmonids, we also hear in Newfoundland of seals coming up the rivers, and there is the assumption, therefore, that they are, in fact, then preying on on salmon and trout. That may or may not be the case. And I appreciate the fact that you can't offer any clarity at this point, but my fear in this is that any discussion is going to result in well, bad policy without good science. Policy without good science is going to be bad policy. As simple as that.
Yep. Yeah, I agree. Yeah, it's a specific. I mean, if seals are in the river, yeah, they could easily be preying on salmonids. I mean, it's something we see in other systems. The trouble or difficulty is quantifying this and how you adapt the sampling of gray seal diet to be reflective on the magnitude of impact that seals can have on anadromous fish population. So that is the current challenge, and how do we make the sampling design to be able to address these questions? And there's lots of logistics involved and things like that. But yeah, definite area of improvement. Yeah.
Thank you.
Thank you.
Thanks, Xavier. We do have a question that's been typed, so I'll just read it out loud for you. So from Monsieur Chaput. First, thank you very much, Xavier. It's a great combination of physiology and telemetry approaches with fascinating results. Based on the captive reared wild study from Cape Breton, it seems that it is not nutritional state, but rather stress responses that motivate early migration out and perhaps lower survival. Is a short-term stress associated with handling and transport, or do you know if the hatchery held fish were stressed throughout their holding period?
Yeah, first of all, hi, Gerald. It's great to hear from you, even if it's not your own voice. But yeah, trying to remember the different sub-questions. But yeah, it is, the hatchery fish were sampled for indices of their nutritional state at only one period, and that was the time just before they were being tagged and being ready to be released. We had planned for pre-sampling these fish as soon as they were captured on the river to have the pre-hatchery and the end of the hatchery confinement period, and compare the stress parameters and immune status. But unfortunately, that didn't work out. So it's hard to say how stress level might vary during these months of captivity. I just, yeah, it's difficult to comment on that one. The other question was related to whether or not it's stress or nutritional indices. I think the two, it's difficult to say whether it's one or the other. I think the two can be connected. And what I mean by that is that nutritional state can be important if you look at wild fish, because those that have that are in poor condition, they just won't be able to afford the extra starving for a few months. So they would have to leave if they have to resume feeding and give them a chance to survive. In terms of the hatchery fish, they're stressed, so I would still think that the nutritional state of these animals would be important. I think the main determinant in that one seemed that, yeah, when they were back in the river, they wanted to get out. So I'm not sure if it's and the fact that they're stressed would also mean that they would consume or they would have consumed their nutritional reserve sooner than just a fish that is not chronically stressed. So yeah, at the moment it's difficult to untangle both. I did, if I remember correctly, have a look at the hatchery fish to see if these early autumn migrant versus the one that still waited for the spring to migrate out were different in relation to nutritional state. And they were not, at least for what I could tell. Yeah.
Thanks, Xavier. There's just one last question and we'll be able to close after this. And this one I can probably answer because it is asking if this is being recorded today. Because I know a lot of folks after always email me and have been wanting to attend and wasn't able to and wants to be able to access and see the presentation. So the answer is yes, it is recorded today and it's going to be available on the Foundation's YouTube page. So please, you know, for those that were not here, they'll be able to go. I do see one hand raised. We could allow for one more question, Xavier, if you're okay with that. Okay, just seconds from Mr. Stiles. Okay, can the attendee push the mute button? Some unmute requests. Okay.
Hello.
There you go.
Do you hear me now?
Yep.
Okay, thank you. I was just interested in the one sea winter salmon or repeat krill spawners as to where they feed. I understand that you do not go to Greenland. It's just a 2 sea winter and multi-sea winter fish. So I was wondering where do the 1 sea winter salmon spawn or repeat krill spawners?
Where they feed. Did I understand correctly? Where the 1 sea winter fish feed is the question?
Yes, it is.
Okay. Yeah, that's actually a good question. It would depend on the population. Some 1 sea winter fish in the Bay of Fundy, for example, would stay within the Bay of Fundy. In terms of the eastern coast of Newfoundland, they would also stay along that coast. In terms of population close to the Gulf region, it is thought that these 1 sea winter fish would feed in the Gulf of St. Lawrence for that year. Exactly where, I don't know. This would be mainly derived from PSAT tagging, so satellite tagging of these animals, which would give complete tracks of where exactly this animal went. And I think actually it must be, I didn't have a look recently, it must be part of some of John Strom work. I'm going back. He has a few paper on, I had it, about, it's in here somewhere, yeah, ocean predation and mortality of adult Atlantic salmon. That paper and scientific report here. I know that some of his work documented some movements of 1 sea winter salmon in the Gulf of St. Lawrence. But in terms of the acoustic telemetry data that I used, it is patchy information in the sense that we can only detect fish passing through an array or a gate of acoustic receivers. And of course, we don't have acoustic receivers everywhere in the Gulf. So we don't generate that type of information from the data that I presented.
Yes, you did have a slide earlier on that showed 1 sea winter fish, not going as far as Greenland, and the 2 sea winter, multi-sea winter fish did go to Greenland. So would I be correct in saying that the repeat growth spawners and one sea winter and maiden fish do not go to Greenland to feed?
Yeah, this is at least what I'm familiar with in these assumptions, and I get the map that you referred to is just a general understanding of where the animals, the 1C winter versus 2C, this one, go to. But yes, it is believed that the 1C winterfish and also the consecutive repeat spawner would have a more local migratory strategy, also just based on the fact that they can be out at sea just a few months and then need to come back to spawn. And in terms of the 2C, 3C winterfish and alternate repeat spawners, these are believed to migrate to the west coast of Greenland. But yeah, I don't have a lot more information about this. Nothing more, at least, that we learned from the work I presented on this aspect.
Okay, thank you very much for that. But I'd just like to clarify a little bit on the maiden fish or 1C winter fish. When they do return to rivers as a large repeat krill spawner, do they feed off locally, or do they actually go as a 2C winter salmon, multi-sea winter salmon to Greenland?
Yeah, I understand more where you're coming from with that. Yeah, so I guess the limitation here is that if you refer to that current study, like we pooled repeat spawners in one category, but of course repeat spawners would be constituted of consecutive spawners, which would have a migratory strategy more familiar with the 1C winterfish, but they would also contain alternate spawners, which would be more the ones that migrate to West Greenland. So if I get your where you're going to right, we would learn a lot more from looking at these trends of iteroparity by dividing up the repeat spawners in alternate versus consecutive repeat spawners. And that would allow to learn more about, because right now the fact that they're pooled could be that, for example, consecutive repeat spawners increased a lot, but not the alternate repeat spawners, or the other way around. So it would inform us about which area might have been more beneficial, or the condition improve in one area but not in the other one. But in that instance, all repeat spawners were summarized or included in the same group, while they might have fairly different migratory strategies feeding at different places.
Thank you very much. I really appreciate your presentation.
Thank you.
Well, that should be the end of our questions and answers. If some didn't get an opportunity to ask a question, you can always send it to me by email, and we can do the best we can to answer them. And of course, like I said a while ago, this is recorded and it is going to be available on our YouTube page. So great presentation, Xavier. I just really appreciate your time today and to be able to share that information with us. Before we do leave, I just want to send a little reminder to everyone that we do have another webinar at the end of this month. It's on November 30th with Jordan Condon on the determination of factors affecting survival from egg to juvenile for Atlantic salmon and PEI. So once again, big thank you to Xavier, and also thank you to everybody that joined us today to participate. And we hope to see you all soon. And thank you. Have a good afternoon.
Thank you very much.